In this video, I will explain the basic concept behind antigen processing and the type of immune response against a particular antigen. So, antigens enter our body all the time, but antigens behave differently and our immune response against each antigen is also different. So, what are the major factors which will be deciding the differences in this immune response against different types of antigens? So there are two main factors which will be deciding what kind of immune response will be there and how these antigens will be processed. The first one is the mode of entry of pathogen, whether the pathogen is entering directly into bloodstream or pathogen is invading our tissues. This will decide where this antigen will be processed. So the point is, if the antigen is entering directly into our blood, it will never be processed in the blood. It will be taken to spleen through blood filtration process and it will be processed inside the spleen. And if the antigen is invading our tissues, it will be taken by antigen presenting cells into the respective lymph nodes and it will be processed inside the lymph nodes. That is the whole point. Now, how this antigen will be processed and what kind of immune response will be there? What kind of cells will be involved in this immune response? How this antigen will be processed and presented? Whether it is presented by MHC-I, major histocompatibility complex 1, or MHC-II. All these processes will be dependent on the nature of antigen. What is the nature of antigen? Whether it is a protein type antigen or it is non-proteinaceous antigen. In case of non-proteinaceous antigen, it can be some kind of lipopolysaccharide. And all these factors contribute to what kind of immune response will be there against this antigen. So, if antigen enters directly into blood, it will be carried to spleen and it will be processed inside the spleen and effective immune response will be initiated. So, now I talked about two factors. The first was mode of infection. We are talking about the infection directly into the blood. And second factor I talked about was nature of antigen. Now, if nature of antigen is protein, both T cells and B cells will respond to this antigen. And it will be taken to this T cell zone as well as these B cell marginal zones. That is for the proteinaceous antigen. But if antigen is not proteinaceous in nature, but if antigen is the non-proteinaceous lipopolysaccharide, then only B cell response will be there and this antigen will be taken to these marginal sinuses where B cell populations reside. That's how the antigens of blood will be taken care of. Now, what if the site of infection is directly in the tissue region? We will talk about this tissue region in detail, whether the type of antigen is extracellular or intracellular, but in both the cases, this will be carried out through lymphatic system and it will be taken to lymph nodes where it will be processed. Just like spleen, the concept is that if the pathogen or if the antigen is non-proteinaceous, only B cell response will be there and it will be carried to these B cell follicles. And if it is proteinaceous antigen, it will be carried to these T cell follicles and both B cells and T cells will respond to this antigen. To summarize this concept, if the site of entry of antigen is blood, it will be carried by blood into the spleen. If the site of entry is tissues, it will enter the lymphatic system and it will be carried to lymph nodes by antigen presenting cells. And if the antigen entry is directly into mucosal surfaces, which are like mucosal associated lymphoid tissue or Peyer's patches, the mechanism of processing will be presentation through dendritic cells. So, this is the summary of the site of entry and processing of antigen. Now, we will talk about these processes in somewhat details. If antigen enters blood in free form, non-processed form, it will go to spleen and there may be B cell response, there may be T cell response. But the nature of antigen will decide what kind of response will be there. If the nature is lipopolysaccharide, there will be only B cell response. So it will be taken down to marginal zone of spleen. We talked about in this figure. marginal sinuses where B cell populations reside. And the second concept we need to know is for lipopolysaccharides there is no class switching. There will be only production of IgM antibodies and there will be no T cell response. T cell response is only carried out against proteinaceous antigens. So proteinaceous antigens will be carried down to white pulp or PALS, which is periarteriolar lymphoid sheath. We talked about in this diagram. This is T cell zone, PALS, where T cell populations reside. So there will be massive T cell response. These T helper cells will migrate to B cell follicles and initiate B cell response. There will be generation of antibodies. There may be class switching and there will be affinity maturation and there will be memory response as well. So what is the sequence of events? Bacterium enters blood, it goes to spleen, there may be complement activation as a part of innate immune response, there may be neutrophil swarms due to respective interleukins, interleukin 8, there will be splenic macrophages, phagocytosis, there may be toll-like receptors activation and their respective responses, there may be inflammatory cytokines will be released, there will be activation of T helper cells and T helper cells will help activation of B cells and these B cells will generate IgM antibodies. The nature of antigen is proteins, there may be class switching and class M will be switched to class G and the bacterium clearance will be observed by the processes like opsonization and phagocytosis. There will be formation of memory cells for these proteinaceous antigens for upcoming secondary infections as well. That's how the antigens of blood will be processed and the nature of the antigen will decide but will be the sequence of events of processing of these antigens. What kind of cells will be involved? For proteinaceous antigen, both T cell as well as B cell will be involved. In case of non-proteinaceous antigens, only B cells will be involved. There will be no class switching because to switch classes we need proteinaceous antigens. Now, if the site of entry of pathogen is tissue, it will enter the lymphatic system and it will be carried to lymph nodes. If the nature of antigen is intracellular, it will be processed by the proteasomal processing system and it will be presented through MHC, major histocompatibility complex 1, and T cytotoxic T cells will respond. CD8 positive T cytotoxic cells will respond and further immune response will be carried out. If the nature of antigen is extracellular, there will be the endosome lysosomal processing, and if there is endosome lysosomal processing, MHC-II presentation will be carried out, and for MHC-II the cells or CD4 which is specific for MHC-II compatibility, the T helper cells will respond and further downstream processing of immune response will be there. These T helper cells may activate B cells, there may be generation of antibodies and other immune responses will be there. Now, in a nutshell, the tissue antigen, if it is extracellular lipopolysaccharide, it will not be processed. MHC processing will not be there. MHC processing is only for the proteinaceous antigens. The APCs will be involved, which are macrophages and dendritic cells. T cell response is only for proteinaceous antigens, so there will be no T cell response in case of extracellular lipopolysaccharides, and there will be only IgM, there will be no class switching. If it is intracellular LPS, there will be no processing, no MHC, no APC, no T cell response, no B cell response. These responses will be absent altogether. So how these intracellular LPS will be processed? There will be no B cell response, there will be no T cell response, there will be no formation of antibodies. So what will be the immune response against the intracellular lipopolysaccharides? So there is activation of caspases. In humans, the caspase 4 and 5 will be activated. In case of mice, there may be caspase 11, and these caspases will cleave a protein which is gasdermin D, and this gasdermin D will cause the cell swelling. The plasma membrane of cell will lose its integrity, and there will be initiation of pyroptosis, which is inflammatory cell death, and it will kill the cell quickly. So If the nature of antigen is intracellular LPS, there will be initiation of pyroptosis, and this initiation of pyroptosis is through this gasdermin D, which is activated by caspase 4 and 5. So there will be massive inflammatory response, and the cells which are seen in this case is neutrophil recruitment. There will be a lot of neutrophil recruitment in this case. So that's about the extracellular and intracellular lipopolysaccharides. Now, what about the proteinaceous antigens? Let's talk about the extracellular proteinaceous antigens. As we already described, extracellular proteinaceous antigens are processed through endosomal lysosomal system, and if it is endosomal lysosomal system, it will be processed through MHC-II, and it will be responded by T helper cells, CD4 positive T helper cells. It may be T helper 1, T helper 2, T helper 17, and the nature of antigen is protein. There will be class switching from IgM to IgG or IgA depending upon the site of infection. Now come to last one. What about intracellular proteins? Intracellular proteins are mostly seen in case of viruses. They will be processed by proteasomal system, and if proteasomal system is involved, they will be processed by MHC-I. They will be presented by MHC-I, and if they are presented by MHC-I, CD8 T cytotoxic cells will be involved, and they will be activated into CTL cells, and there will be antibody production. Now let's summarize these concepts. If the location of entry is blood and the nature of pathogen is LPS, it will be taken down to spleen. There will be no MHC based presentation. There will be only B cell response. The co-stimulation through toll-like receptor 4, MD2 and CD14 pathways. There will be generation of IgM antibodies. There will be no class switching. There will be complement activation, and some inflammatory cytokines will be observed as well. If the nature of antigen is protein, it will be taken out to spleen. Both B and T cells will be involved. If there is antigen processing, there will be endosome lysosome processing. If there is endosome lysosome processing, it will be taken down to MHC-II presentation, and MHC-II presentation is taken care by CD4 positive T helper cells, and there will be class switching of antibodies because these T helper cells will activate B cells as well. B cells generate antibodies. Antibodies will class switch into IgG or IgA depending upon the location of antigen, and there will be affinity maturation. There will be formation of memory B cells as well. Now, second concept, if the antigen, the nature of antigen is intracellular, the nature is lipopolysaccharide, it will be taken care by tissue resident macrophages. There will be no MHC presentation. If it is intracellular, there may be involvement of caspases and gasdermin activation. There will be involvement of pyroptosis and massive inflammatory response. There will be massive neutrophil recruitment we talked about earlier. If the nature is proteinaceous, it will be taken down to the draining lymph nodes. There will be proteasomal processing, and if it is proteasomal processing, there will be MHC-I involvement, and MHC-I is taken care by T cytotoxic CD8 positive T cytotoxic cells, which will be converted into CTL cells, and there will be release of perforins and granozymes which will promote the apoptosis of the cell. There will be release of interferon gamma activation of macrophages will take place. There will be generation of CD8 positive memory T cells as well. Now, if the nature of antigen is extracellular and it is LPS, it will be if the site is blood, it will be taken to spleen. If the site is tissues, it will be taken to lymph nodes. Various cells will be involved. There will be no MHC presentation, and if there is only B cell response, there will be IgM production. No class switching will be there. TLR4 mediated inflammation, complement system activation will be seen. If the nature of antigen is proteinaceous antigen, it will be taken down to the draining lymph nodes, and if it is in blood, it will be taken down to spleen. The type of processing may be endosome lysosomal processing. If it is endosome lysosomal processing, it will be presented by MHC-II itself, taken care by CD4 positive T helper cells, and these T helper cells may generate Th1, Th2 or Th17. There will be B cell activation, and if the nature is protein, there may be class switching to IgG or IgA depending upon location. There will be neutralization of toxins, generation of memory B cells, and there will be long-term immunity as well. Now, let's talk about tissues, and If the nature of antigen is intracellular, in case of tissues, it is LPS, and if it is LPS, it will be taken care by resident macrophages. It will be taken to draining lymph nodes because the site of infection was tissue. It will be carried out through afferent lymphatics into draining lymph nodes. It will be by these antigen presenting cells like macrophages and dendritic cells. There will be no MHC presentation. It is a T cell independent response. There will be only B cell response. TLR4 based inflammatory response is seen. There will be generation of antibodies. No class switching will be there. No memory B cells will be formed because the antigen type is LPS. In case of protein antigens, they will be taken down to draining lymph nodes to local site. Proteasomal processing will be there. They will be taken care by MHC-I based presentation and T cytotoxic CD8 positive cells, and these cells will be converted into CTL based killing cells, and interferon gamma production will be there. Macrophage activation will be there. The antiviral state in neighboring cells will be induced, and there will be generation of memory T cells as well. Now let's come to last one, which is the nature of antigen itself is extracellular and the site of infection was tissues. If the nature is LPS, it will be taken down to the draining lymph nodes of local tissues. There will be no MHC presentation as we saw with previous one, and if there is only B cell activation, TLR4 response will be there. There will be some kind of cytokine generation, complement activation, IgM generation. No memory response will be seen. If the nature of the antigen is protein, it will be taken down to draining lymph nodes and it will be processed by endosome lysosomal processing through MHC-II presentation, taken care by T helper cells, which are CD4 positive cells. It will be converted to different kind of helper cells like T helper 2, which will help in antibody production. T helper 17 helps in the neutrophil response against the extracellular bacteria, and there will be antibody formation by plasma B cells. Class switching will be there for proteinaceous nature, and there will be strong memory cell response. So this was the basic concepts behind the antigen presentation, that how these nature of antigen as well as mode of infection, how these antigens will be processed, and what kind of immune response will be observed.